over again. Now let's talk about the cell division. Okay, you can see the cell over here. He is going through division. That's a knee slapper. Okay, so this cell division, this asexual reproduction cell division, we call mitosis. Okay, so this is mitosis's cell division explained. All right, so make sure you're keeping good notes as I'm going along. If you need a pause through this video, go ahead and pause. Anytime we talk about a cycle, that means it's going around and around and around. It's going through the process over and over and over again. So cell cycle is a regular sequence of growth and division. Growth and division that all cells go through. Okay, so these cells are constantly growing and constantly dividing. They grow, they divide. 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 All right, so here are all the phases that, yes, you will need to know each of these phases, what happens in the phases, and the order, the correct order. So we have interphase, okay? Mitosis and cytokinesis. Those are our big phases. Interphase is where the cells live. They are growing. This is their active phase. Most cells live in the interphase the majority of their time. Then mitosis is the process of dividing. So this is actually all the dividing process. And these four here, prophase, metaphase, anaphase, and telophase, these are mitosis, the phases of mitosis. Okay, and then the last phase is cytokinesis, when it actually finally splits. It cleaves. Okay, it's no longer just one, it's two. All right, so yes, there is information here. And it keeps, again, going around in this cycle. It cycles through, okay? Yes, all the information is here, but you don't have to worry about writing this all down right now. We are going to go over this. So to remember the order, I usually like to make little fun ways for you guys to remember it, for me to remember it. I use this in college, and you can make up your own if you want. That's up to you. If the one I use doesn't work, come up with another one, whatever works for you. So I just take the first letter of each phase. So IMP, MATC, just so I can remember, and so you can remember the order of the phases. So let's take a look at what I came up with. I met purple manatees at the circus. I met purple manatees at the circus. Interphase, mitosis, prophase, metaphase, anaphase, telophase, cytokinesis. Okay? Bada boom, bada bang. Okay, so our first phase that we're going to talk about is interphase. So on your notes, you're going to have different sections to draw what's happening. You can just draw what's here, but I'm actually going to show you what I drew, just so you can see exactly what I'm looking for in your drawings. So interphase is preparing to divide the cell. This happens before the cell division. It doubles in size, it's growing, it's preparing for division. The replication's happening, it's making copies of DNA. Just like when I pass out papers to my students, I make copies of the one paper to give to everyone. I want everybody to have the same information, so I make copies of it. DNA is the information of the cell, so they have to make copies. So it is bracing for division. So let's take a look at how I drew it. So here's how I drew interphase. So initially it starts out small. This is just living, it's happy, it's great, but it needs to double in size. So interphase, what it does is it doubles in size, twice as big. So what I did is I erased everything and I doubled the cell size. So I have my cell membrane that I drew, and then you will see the nucleus form, okay? And then you will see other organelles pop up, those little yellow dots, and we're going to talk about those later. And you'll see the spindle fibers also pop up, the thin green lines, okay? So that's all I really want is just those, the things at the bottom and the nucleus. If you need to pause the video at any point, I'll go back. If you need to pause the video at any point, then you pause it, draw it, use different colors, whatever suits your fancy, okay? Whatever you have on hand. Okay, so now we have interphase, we just talked about that. So our next phase is mitosis. There are 4 phases within the main phase of mitosis, and we're going to talk about each of those. But why do cells go through mitosis? They use it for growth, for growing, like this plant over here is doing, it's growing, and to replace dead or worn out cells. Like if you get a cut or something, you need to replace those cells. Okay, Wolverine here needs to replace those cells. Super healing factor. Okay, he's healing very quickly, as you can see on the scar on his face. He just got cut, he's mad, but not a huge deal. He heals up very quickly. So he's replacing those worn out cells that have been cut off his face. This happens to me all the time. No, it doesn't. All right. So through process of mitosis, it creates 2 new daughter cells that are exact copies of the parents. Again, making exact copies to heal the wound or to grow something, whether it's the plant or maybe you need to grow a new arm. Like this guy. Yeah, that's all I have to say. All right, so prophase. Prophase means like pro, so let's take off the phase. If I just look at pro, that means to go forth or to put forth. Okay, I don't have those on your notes, so if you want to put that in there, you can. So think about words that say proclaim, propel, propulsion, like a boat is propelled or a propeller, proceed, it's going forward. So it's starting the cell division. The DNA in the nucleus starts to condense and coil up to form a rod. You can see in the nucleus here, these X's are forming. Those are the chromosomes. The DNA is coiling up into a chromosome. Each rod doubles and is an exact copy. So at first it just starts out as a single rod, then it doubles, and now it's a double rod, which is a chromosome. Okay, so let's take a look at the drawing that we need to do for prophase. All right, so I just start out with the cell membrane, and you already have that on your paper. And then I want you to draw the nucleus. Again, you can do different colors if you want, whatever shape. It does not have to be exact, but pretty close. And then the chromosomes in here are coiling up. And I just drew a couple X's in here. I did 4. You can do 6. You can do somewhere around that. You can do 5, whatever you decide. And then again, I just did the centromeres and the spindle fibers up at the top. You'll see in a moment. So I'll just go ahead and sketch those up there. And then the green lines will be the spindle fibers. Now, I haven't talked about those, but you will see why those are so important in a moment. Now, spindle fibers aren't a vocab term or anything, but you will need to know what they're doing at a later time. All right, so back to our presentation. So prophase, meaning to go forth, proclaim. It's going forward with the cell division. Each rod is doubling, is making a copy. It's just preparing, going forward. So if I go forward and proclaim something, I'm telling somebody something. So here's a video clip, and here's a picture or two of somebody proclaiming something. So let's take a look. This is a great classic example of someone proclaiming something to a crowd.
I declare bankruptcy!
All right, so I was talking about chromosomes earlier and single and double rods of DNA. So let's take a step back and take a look at chromosomes and chromatids. So a chromosome, as some, is a double rod of condensed DNA. So maybe circle, highlight, underline, whatever helps you. This is a double rod of DNA. So see, this DNA is coiled, 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 coiled until it makes these rods of DNA. So see, the DNA has just coiled up in here. So this whole X right here, this X is a chromosome. So maybe if you want to put an X next to a chromosome, an X, and then what I want you to do for chromatid is just put a line, okay, like a little squiggly line, okay? Just single, because just one side of it, like this blue side, that is a chromatid. This yellow side is a chromatid. But the entire thing is a chromosome. Some. Okay, so if I have some candy, do I have one? No, I have more than one. If I have a chromatid, you only have one. If I have some candy, I have more than one. So I have some chromatids. So chromosome is more than one. Chromatid is one of the identical rods of chromosomes. They are held together by a centromere. That'll be sitting right there. Okay, you don't have to worry about the word centromere, but just know that a chromatid is a single rod. Okay. Metaphase is the next phase we're talking about. Meta means along with. Okay. Metaphase, a great way to remember this, maybe write this somewhere in your notes, add this somewhere over here, is that all the chromosomes are lining up in the center or in the middle. Metaphase, middle, metaphase, middle, metaphase. The chromosomes are in the middle of the cell. The chromosomes line up across the center of the cell, and these little strings here that I was telling you about earlier, the spindle fibers, they attach to each chromosome at the centromere. So that is that thing right in the middle where I'm putting my red dot. Okay, see the spindle fibers are connected right there. Okay, so when you draw this, make sure you have them connect at the spindle fibers. So again, I'm drawing my circle, my cell membrane, and so I have where the spindle fiber is attached, the anchors. I'm not going to get into another organelle for you to memorize, but that's just where they anchor down. And so here I have my chromosomes lining up in the middle, and the spindle fibers are attaching at the middle of the chromosome. And they're going to eventually pull those chromosomes apart into chromatids. Okay, so go ahead and make sure you have that drawn. You can pause the video if you need it. So that's why I'm going to just keep moving on. Okay, chromosomes line up across the center of the cell. The spindle fibers attach to each chromosome at the centromere. We'd already talked about that. Okay, these stones are lined up. They're very meta. Okay, next phase is anaphase. Ana means against or back. Think of anarchy. Anarchy means you're against the government. So what happens is, remember we had those X's in the center? Well, those spindle fibers pulled apart the chromosomes, and now they are chromatids. They are single lines. They are single rods. So the chromatids separate and move to the opposite ends of the cell. They were together, but now they separate. They are opposite ends of the cell. You can kind of see how there's two cells now forming. The cell is stretched out and is pulled apart. So again, if you can pause the video, go back a little bit if you need to. So that was the anaphase. Okay, so here I have it drawn, stretched out a little bit, and here I have my chromatids being pulled apart. You can see how the force is just too much for them, and they're just being ripped to pieces. Okay, so the cell's just being divided by these spindle fibers that are pulling them apart. And here comes the spindle fibers. I will be checking your notes, so make sure that you have these down, and it doesn't have to be colored or anything, but I will be checking to make sure you have these done. So please make sure you use your time wisely. Anaphase, they're stretching, so I showed a picture of Stretch Armstrong. If you don't know what he is, I think he's making a comeback, but he's a toy that just stretched. It's really like, that's what he did right there. You can see it. That's the toy. Not sure why he's in a car. Maybe he's holding the door shut so intruders don't get in. I don't know. Maybe he's like a lock. All right, telophase is the next one. Telo meaning far or far off. So that's where we get our word like telephone, telegram. Something is far away. Tele, I mean talking to somebody far away. So the reason why it's called tele is because these cells are getting further and further away. So draw a picture of a phone or something, whatever helps you remember that. The chromosomes are stretching apart. They don't look like rods anymore. They're going to start forming the nuclear membrane. That is the big part I want you to know, is that there's a new nuclear membrane formed around each area. So circle, highlight that. That will be on the test. Hint, hint, wink, wink, nudge, nudge. So you can draw a picture of a phone. I did some people doing phone stuff with those cans and the string, which kind of works if you've ever tried it. Okay, let's take a look at how I drew that in telophase. You can see that they're being pulled apart. They're being stretched here. Some stretch marks here. So you got the nucleus, you get the new nuclear membrane being formed, and I'm going to put my chromatids in there. And that's pretty much it. And then I just drew the arrows to indicate that they're being stretched even more apart. All right. Again, if you need to pause it, you can pause it. But I gotta keep moving on. All right. So there's our telephones again. Cytokinesis is our last phase. Cyto just means of a cell. Kinesis reminds me of physical science, like kinetic energy, movement, motion. So an animal cell, we're going to do one for animal cell, and then we're going to do plant cell. So in an animal cell, you can see here, they have split. The new membrane, or I'm sorry, not the new, the cell membrane pinches to form two new cells. Each cell is an identical chromosomes, has identical chromosomes and half the organelles. Now what's going to happen is the organelles will then form the nucleus will tell, the cell, when to make or how to make them using different proteins. So it will get the organelles back. It has to make them because it just was split in half. So it will get them back, promise. All right, so animal cell, they split up completely, but in a plant cell, they do something different. A cell plate forms across the middle of the cell. So this plate just kind of goes across, so everything kind of splits apart still, but there's a cell plate that comes across, and then new membranes develop on the inside, and then that cell plate then turns into a cell wall. So let's take a look at how I drew each one of those. So let's look at just cytokinesis with a an animal cell. Again, you just got the nuclear membranes and then two nucleus, and they've been split apart. That's really it. Okay, I'll play that again. It's just 7 seconds. So I just have them split apart, and that's really it. They're apart. Kinesis, kinetic. Maybe write kinetic energy somewhere on there. Okay, so cell walls. You can see that there. My nuclear membranes. Now remember, cells in a plant are like bricks. So these bricks just keep getting stacked and stacked and stacked. Think about whenever you have a plant, it grows taller and taller. So I have my cell plate that forms across here. Then the cell plate then forms a cell wall. So then I'll draw something that says that this is a cell wall. A cell plate. And then that cell plate will then turn into the cell wall. It'll be a new cell wall. And I'm only telling you that because it's kind of hard to